diagram is about negative prokaryotic gene regulation, and specifically you would find this in like E. coli bacteria or basically any prokaryotic cell that needs tryptophan to survive. And essentially, TRP repressor is coded from TRPR, which is another gene previously that's being encoded regardless. And this TRP repressor will actually act as is initially inactive, such that this gene will be transcribed because it's inactive. But it can be activated when there's a large amount of tryptophan. Therefore, it acts as a negative feedback system, meaning if there's a large amount of tryptophan inside the cell, then it will start binding to this TRP repressor and activating it, and then this activated TRP repressor will then bind to this binding site, and then it'll this operator, and then it'll actually stop the transcription of making more tryptophan, which would be unnecessary, and it would be a waste of energy, and it basically limits the amount of work that cell can do. And so, yeah, that's it. Really. This is another example of prokaryotic negative gene regulation. So in this case, just like a TRP repressor, an inactive repressor is coded previously beforehand by the LAC1 gene, which is as I said, transcribed into this protein. And this protein, initially, this entire thing is actually inactive. This repressor keeps these enzymes from being coded. Now, specifically, what these enzymes do is that B-galactase actually degrades lactose, and trans-aspartase also degrades lactose in order to harvest its energy. And permease actually acts as actually transports this energy and this lactose into the cell. So why would you need this? Well, you need this when there's a high amount of lactose inside the cell and you need to get rid of it. So when there's a high amount of lactose in the cell, fragments of that lactose called allolactose actually binds to the repressor site, which at this point is still repressing this gene from encoding, and it'll actually inactivate it so that it changes shape, just like an allocentric inhibitor. And so when this happens, this repressor, this comes off, and there's no need for this. These genes are coded continuously, and then the lactose is able to be broken down into energy, which can be then harvested by the cell. And eventually, you'll get to the point where there'll be not enough lactose in order to bind to this repressor. So when there's not enough lactose to bind to this receptor, to this inactive repressor, then the inactive repressor turns back to its previously active repressor formation, and then it'll bind back into the operator and therefore stop the harvesting of energy from lactose. And that's how it works as a negative control system. Figure 18.5, which is basically an extension of 18.4. So this diagram that I'm going to be talking about is actually the CAMP diagram, which is talking about positive gene regulation. And so what happens in positive gene regulation in prokaryotic cells is that there's a high amount of lactose inside the cell. And when there's a high amount of lactose inside the cell, there's a low amount of CAMP. And when there's a there's a high amount of CAMP, sorry. And when there's a high amount of CAMP, it's able to bind to an activator protein called CAP. And when this activator protein is activated, then it actually binds to this RNA polymerase, and it speeds up transcription of these genes. Keep in mind that this repressor is already inactive because of the amount of lactose inside the cell, as I said in the previous video. And so when this activated process happens faster, B-galactose and trans-acetylase is able to break down the lactose faster, and the permease is able to transport this energy faster. Therefore, the lactose concentration decreases. So eventually we get to the point where lactose concentration is so low that the CAMP production will also be simultaneously low. And when that happens, that means this entire process stops. This CAMP will no longer exist, this activator protein will no longer be activated, and therefore this RNA polymerase will stop going as fast. And then lastly, obviously, the inactive repressor will become activated again because of the lack of allolactase. That's basically positive regulation. Yeah, so a slight addition to this is that when there's a high amount of glucose and there's a high amount of lactose, there actually is no need for then there's no need to harvest energy from lactose because glucose has a much higher energy content. Therefore, it's more preferable to break down. So this process will occur at the same rate as regularly without this activator protein because the camp levels will still be low because of the high amounts of glucose and the high amounts of lactose. And yeah, that's basically it. Okay, so this is a spliceosome, which basically splices out the unnecessary code that's inside of the pre-mRNA and it splices together exons. So spliceosomes have small RNAs inside of it that actually help it bind to the nucleic acids inside of the pre-mRNA and break down the introns. So, for example, here, this intron right here, the nucleic acids over in this RNA strand right here, and inside the intron itself would interact and they would break down, and it definitely happens on the ends, and it happens sometimes in between the key parts in order to break down the intron, and then the intron is released into this curled intron, and then it rapidly degrades, and it's unnecessary. And exons are then spliced together by the spliceosome somehow, and then a poly A tail is added onto the newly formed mRNA, which helps the mRNA travel outside the nucleus, and the prime cap is added, which essentially keeps the mRNA from degrading. So yeah, that's why cells. So this is all about eukaryotic gene expression. And so first in eukaryotic cells, there's chromatin, which is densely packed DNA. And in order to unpack this, something occurs called acetylation, which loosens the DNA in order for it to be transcribed. And so this DNA is then transcribed into pre-mRNA, which then has to be spliced in order to get useful mRNA out. So the introns are taken out. And then after the RNA is processed, then you have to add a poly A tail, which will help it leave the nucleus, which will help the mRNA leave the nucleus, and also a prime cap, which keeps the mRNA from degrading. And then after that, it leaves the nucleus, and then it's translated into protein, polypeptides, and then the polypeptides either can come together in groups and make useful protein, or by itself make protein. And that active protein can either be degraded after its use, can be degraded, or later on be transported to other cellular destinations.